Okay, welcome to this ARRB LEN seminar series video focused on the genetics research node of the ARRB Research Network. The Autism Intervention Research Network on Physical Health, or ARRB, establishes and maintains an interdisciplinary, multi-center research network for scientific collaboration and infrastructure to increase the life expectancy and quality of life for autistic individuals, particularly for underserved and vulnerable populations. The purpose of the ARRB is to support innovative life course intervention research that promotes optimal health and well-being of autistic individuals across the lifespan. So this video today is going to focus on the ARRB Genetics Research Node. The goal of the Genetics Research Node, the goals are to promote access to genetic testing among individuals with rare genetic diagnoses, particularly among underserved populations, in an effort to enhance physical health in this population. My name is Gabriel Dichter, and I will be the facilitator of this video. I'm a professor of psychiatry at the UNC Chapel Hill School of Medicine, and I'm also training faculty for the North Carolina LEN program at the Carolina Institute for Developmental Disabilities at UNC. I am a white male with short hair. I'm wearing a purple shirt, and my Zoom background is the ARRB logo today. I am joined by the ARRB Genetics Node leader, Dr. Julian Martinez Agosto, Associate Professor of Human Genetics, Pediatrics, and Psychiatry at UCLA. I'm also joined by Zachary Williams, an autism researcher with a background in quantitative psychology, psychiatry, neuroscience, and data science. He is an MD, PhD candidate in neuroscience and hearing and speech sciences at Vanderbilt University. And during our panel discussion, we'll be joined by Ms. Felicia Williams, a North Carolina LEN graduate, a certified community health worker and educator, and connection and resource specialist for the Autism Society of North Carolina, and the parent of a child with autism. Our goals for today are to address the following learning objectives. Number one, to understand why genetic testing for autistic individuals and their families is important. Number two, to understand the importance of taking a lifespan approach when considering genetic testing for autistic individuals. And number three, to understand important ethical considerations related to genetic testing for autistic individuals. And now, to provide a general overview of the ARRB Genetics Research Node, I will turn it over to Dr. Martinez, who is the ARRB Genetics Node leader. Dr. Martinez, would you please start by briefly introducing and describing yourself?
I'm Julian Martinez. I am the ARRB Genetics Node lead. I am a Latino male with no hair, who's bald, who's wearing a checkered blue shirt. And I'm delighted to have the opportunity to give you an overview of how we're using genetics to improve the health of autistic individuals. First, we want to provide a background about genetics and what is the evidence that a condition or a clinical presentation has some genetic contribution. And we know a lot of that from twin and familial studies, where we've documented that the likelihood that there will be a predisposition for having a neurodevelopmental difference can be as high as 90% in monozygotic twins versus much lower when twins are not genetically identical, so essentially not sharing the same identical genetic sequence. So that to us is one of the main criteria for deciding whether genetic testing is something that's going to be useful if we have some previous evidence that in individuals who share the same genetic instructions, you're going to see a higher incidence of shared symptoms. Next slide. So when we're talking about autistic individuals, there can be different types of clinical presentations. You can see autistic individuals that present with no coexisting clinical findings, and then those who present with a recognizable pattern of associated both physical and clinical findings that affect many different organs that can then suggest a particular genetic predisposition. Next slide. So for those individuals, the goal of a genetic evaluation is to try to make an impact. And this will address a lot of the topics that we're going to discuss today, which is to try to better understand the clinical course or the natural history of genetic predispositions and how they manifest in both physical health and general health. The ability for individuals who want to have access or should be pursuing genetic testing to reach out to community physician practices, to be aware of what a genetic evaluation entails, be aware of practitioners that facilitate that process, and how to involve their families in decision-making as to whether that is the most appropriate next step. The inclusion of genetic counseling in the whole process to make sure that the diagnostic journey or odyssey that many patients go through, stumbling along the way through the medical system, trying to identify a particular cause that can bring together understanding and perspective as to why has someone been experiencing a myriad of genetic symptoms or physical findings that together tend to happen more often when they're associated with a genetic predisposition. And making sure that as along the process, there's proper counseling before genetic testing about what it entails, its risks and benefits, and also what the clinical utility is once pursued. Of course, the intent of genetic evaluations and testing is to anticipate complications. Many times individuals with predispositions will have co-associated clinical findings like cancer and seizures that could benefit from early diagnosis and treatment. Of course, there's a role for genetic testing in family planning. We have many instances in which more than one individual is affected with multiple medical concerns in a family. And that can happen before even a diagnosis is achieved. And then the reason that multiple children are affected could be because they all share the same genetic predisposition. Of course, that then leads to potentially being able to identify affected relatives or other family members that might also be carrying the same genetic predisposition, and that will then allow, through genetic cascade testing, we call it, identify other individuals that would benefit from surveillance. And the intent here is to identify diagnosis early so that we can have better outcomes with early interventions. So then that brings me to the next step, which is what is the usefulness or the utility of doing genetic testing? And the main reason is to try to develop a personalized treatment plan for each individual by identifying, once the diagnosis is achieved, what is going to happen later on as the individual progresses in their lifespan from childhood all the way to adulthood. What are we expecting to occur that could affect their health? Take advantage of existing either therapies or interventions that we know might be very specifically effective in the individual's diagnosis. And then, of course, if there are no available interventions currently, the opportunity to participate in future intervention trials that could potentially be useful for that particular genetic predisposition. Next slide. So as part of the genetic evaluation, we seek to integrate several components to try to identify if there is a family history or a predisposition. So looking at the symptoms and clinical presentation of other family members is a very important clue. Understanding what happened during the pregnancy is also important to tease apart, are there what we call teratogenic effects? Are there things that environmentally might have affected development of the individual during the pregnancy versus something that's intrinsically genetic? Once the individual is born, understanding their developmental history, what were the differences that were noticed as individual milestones in development were achieved? And of course, pursuing a physical exam to find differences on physical exam that could be clues to other internal differences that might be pointing to improvements in interventions on health. And then, of course, we achieve all of this through genetic diagnostics testing, and I'll give you a flavor today of some of those examples to put it in proper perspective. Next slide. So what exactly are we trying to identify here? We're trying to identify differences in our genes, and genes are the instructions that tell our bodies how to function. Genes essentially make a message that is turned into proteins, and proteins are the deal makers in the body. Essentially, they're responsible for every function that allows our organs and our bodies to thrive and be healthy. And of course, the genetic material is packed very tightly into each cell to ensure that it's protected and it actually is turned on and off whenever necessary. Next slide. So The way that it's packed is in what we call little chromosomes, and they look like little spaghetti strands. We have 2 pairs of 22 chromosomes. One copy of each chromosome comes from each parent. So for chromosome 1, you have 1 copy of chromosome 1 from mom and 1 from dad. Same thing for chromosome 2 and all the way to the 22nd pair. And then the 23rd pair of chromosomes makes us boys or girls, right? So essentially X and Y gives you a Y karyotype, which essentially gives you sort of certain developmental pathways, and then XX gives you a different developmental pathway. Next slide. So when we're looking at genetic information, we're looking at differences, and those differences we call genetic variants. And those could be differences in the number of genes, right? We all have 2 copies of every gene, 1 from mom and 1 from dad. And sometimes 1 or more copies of a gene can be missing or be there in more copies than normally expected. Or there can be a spelling change in the instructions of a gene. And you can imagine that a gene is almost like a recipe. And if you have a recipe for making a cake, and instead of saying 1 cup of sugar as part of the recipe, and instead it says 1 cup of sugar, right? Just changing that single letter can change the meaning of the recipe, right? Baking a cake with a cup of sugar versus a cup doesn't allow it to come out quite right. So the same thing can happen to genes. Changing 1 letter can alter the function of the protein and therefore cause downstream effects on our health. Now, when we look at those genetic variants, they could be acquired, meaning that they only happen in a particular group of cells or tissues in your body. And those are usually limited to those tissues. Those are the things that, for example, lead to excessive growth of cells, which can lead to cancer, things that happen unexpectedly because there has been some spelling change as cells grow that have happened either because of exposures to environmental agents, viruses, or things like that, that our bodies usually repair by having DNA repair mechanisms, but sometimes those go missed and then lead to unexpected health changes. And those are different from inherited variants, which are changes that you're born with, that are present in all of the cells of your body that then predispose to having differences in the function of specific organs, depending on whether the function of the gene is required there or not. Next slide. So I'll give you an example of one of those changes. So one is a copy number variation, we call them, which in this example is a deletion. So for example, if you're making eggs and sperm, you're passing down those instructions to make the baby in the future, you can see that on the paternal side, which I'm using here blue just for the purposes of illustration as being the paternal line, there's a piece of that genetic material that's missing. So when you go on to the next generation, you'll see that that actually gets passed down as a loss of genetic material, and then that can lead to specific symptoms. Next slide. So the technology or the test that we use to detect whether there's any missing copies of genes is called chromosomal microarray testing. And it's essentially just a computer chip where there are neighborhoods of genes that are represented by each one of those yellow dots that you see there. And what you do is you take the individual's genetic material and then you essentially allow it to stick to that chip. And that what the chip spits out is essentially a diagram of neighborhoods where genes might be missing or being present in extra copies. So it's a very effective way of looking at all of our 20,000 genes in one test and figure out if any of them are in 1 copy or in more than 2 copies. Next slide. Now that's different from, for example, sporadic mutations. So for example, in this case, you're making sperm where the star is telling us that there's a genetic change or a spelling change in an instruction of a gene, and that gets passed down to the next generation, causing situations. The reason that it's spontaneous or sporadic is that the change isn't coming from either parent. It's just happening spontaneously as the sperm and the egg are being made. And that then gets passed down to the next generation. We usually tend to call those de novo or spontaneous changes, and they're very common as the mechanism for why these genetic predispositions occur. Next slide. And that is very different when you have an inherited familial mutation, where again, the sperm in this example is passing down the genetic change, but it's because, for example, the father in this case is carrying that genetic change. And that's why it's being passed down, right? So those are what will be called familial or inherited genetic changes. Next slide. So how do we assess whether there have been those spelling changes? So there are different tests, and each test has different numbers of genes. The most comprehensive one that we use is called whole exome sequencing. And we recently had guidelines from the American College of Medical Genetics that were evidence-based that have identified whole exome sequences as either a first-line or second-line test that is very comprehensive to assess whether there are spelling changes in those instructions of each gene. It does it for all 20,000 genes. But what it does is it focuses only on what are called exons, which, as you can see here, DNA is made of exons and introns. Exons are the pieces of the DNA of the gene that are spliced together or sliced together to make a functioning message that then gets made into a protein. So by definition, the exons are the ones that code for the proteins. So if you're going to have an effect on the health of the individual, it will be because the protein is different. So therefore, the parts of the gene that make the protein, the exons, is where the changes more likely will occur. And that's essentially what the test does. Now, mind you, of our genetic material, that only represents 1%. The other 99% is introns and what we used to call junk DNA, but now we realize that it actually has important roles in controlling when genes are expressed and how. Next slide. So that's in contrast to whole genome sequencing, which is a test that is just recently being translated into clinical care, where there you're not only looking at the exons, but also at the intron sequences. So now you're looking at 97% of the genetic material. We now know that when you now look at the whole genetic material, or at least most of it, 97% of it, and look for genetic changes, that you can have even more higher likelihood of finding a genetic change, in this case, up to 40%, of finding a change that might be hiding in those introns that are responsible for putting together the instructions that make proteins, right? So one test is more comprehensive than the other, and one can lead to more diagnostic yield than the other. Next slide. And then to summarize, essentially all these types of tests give us different yields because we know that the causes or the predispositions can come in different flavors, as I've introduced to you. And about 20% of the time, there are those copy number variations or missing genes. The remainder are a combination of having a spontaneous change in the gene or having a change that's inherited from both parents. But the remainder, which is about 55% or so, we still don't understand if there is a genetic predisposition or not. Many hypothesize that those are just common changes that we all carry, that in combination or a particular pattern may increase the risk of having a specific health concern, right? Or a developmental difference. Or are there other mechanisms for how genetics can lead to a predisposition to health differences that we have yet to identify? So what this all brings as part of the testing is the question of when to do it and whether it's actually appropriate to do it. Just because the testing is available, does that mean that it's appropriate or adequate? And one of the things that we're going to discuss today is the ethics of in which circumstances is genetic testing most appropriate, and in which ones we're still having a conversation about how that genetic testing should look like. And of course, how research is allowing participants, autistic individuals, to participate in genetic studies, and how can we steward their participation, protecting their rights to that information, and how they actually decide that information is used.
Thank you, Dr. Martinez. So we are now going to transition into our panel discussion. And over the next 20 minutes or so, we're going to cover 4 topics. So now we're going to be joined by Zach Williams and Felicia Williams. And would you two please briefly introduce and describe yourselves, starting with Zach?
Sure. Hi, I'm Zach Williams. I'm a 6th-year MD PhD candidate at Vanderbilt University School of Medicine. And I am also an autistic self-advocate. And so I'll be providing a personal experience as an autistic person, as well as an autism researcher for this panel. And physically, I am a white Hispanic young adult, and I have black hair. And my background is a white background with the ARRB logo in the top right corner.
Great, thank you, Zach. And Felicia, please.
Yes, my name is Felicia Williams, and I am, I guess, a middle-aged Black woman with long hair. I'm wearing a coral dress and a colorful scarf. My background is white with the ARRB logo. I am a connection and resource specialist with the Autism Society of North Carolina. I've also done a lot of self-advocate work as a community health worker. And I am also a mother of a child with autism. He is 13 years old. And I am here representing the parents. Thank you.
Great, thank you both. So our first topic is to discuss and talk about parent versus provider versus personal perspectives on genetic testing. And so if Julian, if you wouldn't mind leading us off on sort of your views on these different perspectives, and then we'll sort of go through the group, please.
I mean, I think as a provider, I've essentially discussed or set the background for why we feel as providers of genetic testing and genetic evaluations that the opportunity to pursue them is helpful for individuals, right? Whether they're children or adults, autistic individuals. Now, what I have experienced as a provider is when you interact with families and autistic individuals, that perspectives can be very different, right? And one of the ones that we find a lot is concerns about how that information is stewarded, right? What are the implications of that information on insurability for certain individuals, depending on what those findings look like? And then, of course, when it comes to making a very personal decision about whether this is important in the family dynamic, right? One of them is this idea of if it's a genetic change that's inherited from one parent, what is the impact of that on the family structure? About questions of, was this passed down by you, and this is why so-and-so is like this or that? And then questions about perspective on pregnancy and motherhood, right? And realizing what could have happened, what could I have done, could this have been different, right? And sometimes the genetics evaluation provides perspective as to the fact that there was nothing that happened in the pregnancy that could have been any different, and how that sometimes the diagnosis can put some perspective on that uncertainty, which a lot of parents carry about, could it have been different? Could it have been something that I ate? Could it have been some falling during the pregnancy? Could it have been the evil eye? All these different things that we hear all the time, and lifting some of that concern away.
And before moving on to Zach, could I just follow up, Julian, in your experience, roughly what proportion of families when offered genetic testing are interested versus resistant to it?
Yeah, I mean, I think there's two corollaries to that question, right? Because first is how many families who are offered by their referring physician, right, to come actually come. And it's actually a very small number. So I hear from my primary care colleagues that even though they offer the referral, very few actually follow through, right? It's usually about 10%. And that's something that I think it's a gap that we want to address. What are the concerns at that point that are not being communicated, that are leading those families to perhaps not pursue it? And it may be perceptions about futility, about, is this really going to help? Why am I doing this? Why is this important, right? So I think that might be an important gap to fill. And then that's different from the ones that actually do make it to the genetics appointment. And then we offer it, and then they say, well, let me think about it. And a lot of that, I think, has to do with family dynamics, number one, right? Are parents on the same page about testing, or is the individual ready to pursue testing based on the risks associated with it through the counseling that we do pre-testing? But also, I think it has to do with costs, right? Because a lot of the times insurance don't cover it, or there's a high copay or co-insurance that you have to fulfill. And then the third one is the question that I always get, which is, why are we going to do this? And many times the argument that we make is not good enough, right? And I think that it's perfectly reasonable to say no and move away and step away and say, Dr. Martinez, I'll wait until we have more opportunities for treatments or something that's more meaningful to whatever is important to that family, right? Because that will be different to different families. Some families are information seeking, they want to know, and they want to have the opportunity to take advantage of what we have to offer. But for many families, that is not the case, and that's perfectly fine for us.
Thank you very much. Zach, what's your perspective on this topic?
Sure. And I think, just as Dr. Martinez was saying, I think that there's a sort of a very different group of perspectives among the personal perspectives. So, I mean, I have actually had my own genome sequence, not clinically, but as part of participating in the Simons Foundation Spark study. So it was a research study, and they did a whole exome for free. Now, I guess as full disclosure, I got no like pathologic findings back. Apparently, the yield in Spark for participants that they've sent things back on is about like 8%. And so I'm part of the 90-some% who got, nothing of interest back. But that's also fine, I suppose, because I at least got some information about my genetics, and it was clinically interesting to me as a scientist. But anyway, to think about that, one thing to notice for the autistic community is there's a bit of distrust in the entirety of the research enterprise, and genetics in particular has been quite a hot topic lately. In fact, there was a recent study of autism genetics in the UK called Spectrum 10K that was a particularly like hot button issue within the greater autism community, and sort of made it out into even like the lay media about this, which was a study of about 10,000 autistic people in the United Kingdom, and the goal was to basically get genetic samples and associate them with all sorts of health conditions and different things. But basically, there were all sorts of issues with the way that the study was described, both to the participants and then in the grant to the funders, and how those things were disconnected. And people felt like the researchers weren't being honest, or they weren't necessarily putting in the right protections for the genetic data because they were hoping to bank them and allow people to reuse them. But like what they may have been reused for could have been malicious, or things that the autistic community didn't want them to be used for, but we weren't sure that the right protections were in place, et cetera, et cetera. Anyway, so all of this to say is there are many, many different issues that lead, the average autistic adult to be somewhat mistrustful of your the autism research infrastructure, and especially when things come to genetics. Now, I think that families of autistic individuals, especially those who are newly diagnosed and have a child who has significant developmental difficulties and are seeking more services, may be more amenable to a more medical approach and information seeking. I think if you have a newly overwhelmed parent who has a diagnosis of a child and is willing to get that coming in, then they're probably very happy to seek diagnosis. But especially for like later diagnosed autistic adults, I think very few of them go in and seek diagnostic testing. Me getting this through Spark would have been the only reason I would have ever done it. I don't feel like I mean likely a low yield anyway, but had I gone to my psychiatrist, I would not have paid even under insurance to go and get a chromosomal microarray and Fragile X or whatever they're recommending for me these days. And so, right, I think that the yield, especially in adults, is also something that's not really evidence-based. All of the recommendations are at the time of first diagnosis, which is really looking at pediatric samples. And so other more nuanced scientific issues may play into this too, even though that may not really be what your average person is thinking when they walk into a genetics clinic. But again, a lot of different perspectives and a lot of different issues, and happy to talk about this more as we move through the panel.
Great, thank you, Zach. And so Felicia, as a parent, I'm very interested on your thoughts on this topic.
Yeah, well, first, I totally can relate to what Zach is saying. From a parent perspective, you are concerned about transparency, how that information will be used, and is it really beneficial, or is it going to create more anxiety and fear? So that's definitely something that, as a parent coming from a parent's perspective, I would be concerned about that for my child. I did want to say, from a parent to the medical side, it makes me question, how do you determine who is given the opportunity? How is it offered? Like, is it offered to just everyone, or is it offered to families who already have a child with autism? You know, how, I guess, how do you determine who is offered the genetic testing? And then are the providers that are doing this, are they serving a diverse community? I think that is important because I know you guys said 10% actually do it, and I know there's a cost barrier, but I think it's important to note that as a parent, I would like to know, no matter what area I'm in, I have the same opportunity to get this information if I was interested in that. So from a parent's perspective, that would be something I would think about, but also just the fear. For me, what comes up is it's nice to know because then it will affirm, I guess, some of the concerns that you have, but then the flip side of that is, okay, will this negatively impact my child? And will this help with us accessing services? Because it's so hard right now. The medical system has one qualification criteria, the school system has another. How does the genetic component falls into that, and how will that be utilized? So it just opens up a whole nother door to how will this information be received by the community. So yeah.
Those are great perspectives, and it's a really nice segue into our next topic, which is, should every family with an autistic child or adult be offered genetic testing? And we'll start with you again, Dr. Martinez, please.
So I think to address Felicia's comments, which were right on target, what we use as genetic providers as a criterion for offering testing is twofold. One is the autistic individual experiencing other co-associated physical findings, right, or clinical concerns, right, that are impacting their health, right? And I think that's a very important criterion because based on the research that we have, that is the population that will most likely benefit from testing, right? If you're an autistic individual who's otherwise healthy and has no other major clinical concerns or physical differences, the likelihood that the genetic testing, at least as we use it today and how it's intended to be used, will be very low yield, as Zach explained earlier. So I think it's important that, as Zach said, participation in research studies that are trying to understand why is there such a discrepancy, right? And differences, and what it may be the genetic reasons behind that is useful. And I think that's why participation in studies that are trying to understand that is going to be important. But I think it creates a difference in referral and who should be evaluated, right? Now, we don't put the burden on deciding that to the families, right? What we want to do is educate primary care providers to recognize who are the individuals that would benefit most and then refer them, right? So that they can then access to the genetic testing as soon as possible. Now, having said that, genetics is very humbling because most of the time, even though we think we have all the answers, we always get proven wrong. And I've been proven wrong many times where I've had individuals where I felt the likelihood of genetic testing was going to be extremely low, and parents are very big advocates for pursuing it. So we pursue it, and we actually find a genetic predisposition simply because genetic predisposition is so variable in many cases that you could have someone who carries it, but otherwise is completely healthy. And I think understanding that variability is where those other genetic testing, the other genetic research studies are going to give us insights into why is it that individuals that have changes in the same gene present so differently, and in some cases with no health concerns at all. So stay tuned, because I think as more participation in these questions and these studies occurs, we'll get more information about why that happens. And then I just wanted to follow that with Felicia's concern about the anxiety, about the fact that even if you offer genetic testing and you do get a diagnosis, Then you transitioned from the diagnostic odyssey to the post-diagnosis odyssey, which I call it the post-diagnosis PTSD. And that is the combination of the anxiety or the what ifs or the hypervigilance about surveillance, making sure is this going to happen or not, right? About the what is going to come in next, what's new, what can I know, right? What can I do that is not yet to be found to help? And I think that creates its own sort of different state, right? Initially, it's the what happened, but then the after is, now that I know what happens, what can we do about it? And that comes with its own burdens as well, absolutely.
Zach, what are your thoughts on whether everyone should be offered genetic testing who has an autism diagnosis?
Sure. And it's a really interesting question. And I actually think that there are different ways to go with this. You know, to some extent, my answer really is no. If the you know, genetic testing means should everyone get like a whole exome or a whole genome sequencing, you know, yes, you could probably find a lot more and a lot more people by doing this, but right now I think that, you know, if as a medical system we wouldn't really be able to shoulder the costs or make the interventions that we have based on those findings worthwhile. But for the big syndromes that we have, you know, based on copy number variants, because those things have been around longer and studied more, I think that, you know, based on current recommendations to do like a chromosomal microarray and Fragile X testing, based on just that and the costs of those things, we could probably justify at least all children who are newly diagnosed getting offered a test, and then perhaps even tiering that to say, like, you know, if there are additional dysmorphologies or other physical findings that would increase the likelihood to have that be more strongly recommended. It would be, you know, I do appreciate the opportunity to have that offered universally, because I think it does cut access barriers to put additional you know, guidelines on that. That being said, you know, I do still think currently, you know, you're not going to get more than 20% yield with these tools if we screen everyone. And so you know, there may or may not be justification cost wise to do that. And if we stratify, we might be losing some kids, but it could be better overall. So I don't know. Again, you know, it's a question for health economists, but I think that there are certainly reasons to go, you know, both ways.
Great, thanks, Zach. Do you have anything else to add to this topic, Felicia?
Yeah, I would just say I think that for someone who is questioning, you know, they have not received an evaluation, they're noticing some of the symptoms. It would be nice if there was a way that they can inquire about it. As far as it being offered to everyone, I don't think just right off the cuff, no. I think there, of course, like you guys are saying, there should be some concern or some qualifications, the primary care, or, you know, a family already has a child with autism, something of that sort, just because of the anxiety it produces. And then I think for those who do it, there should be some form of robust counseling and transparency of all of these risks and concerns, how this will impact a family, how this will impact an individual. So yes.
Great, thank you, Felicia. We have two more topics. The next one is to discuss what the ethical concerns associated with genetic data and what can be done to address them. And that could refer to clinical genetic testing, or as Zach was talking about, research genetic data and testing. So Dr. Martinez, do you want to start us off?
Yeah, I mean, I think this is a very broad topic, so we'll just sort of touch very superficially the main concerns. So I think on the clinical side, the concern is always the fact that the research is moving faster than the conversation about whether the things that we can do should be done or translated into clinical care or clinical diagnostics. And I think one of the emerging one is for individuals that might not have sort of a single gene or gene loss associated predisposition, but more of the common genetic changes that kind of come together to perhaps increase that risk. meaning multiple changes in many different genes that in combination then increase the likelihood of having health concerns. That if there's actually a mechanism or a tool that allows you to create a score or some kind of guidance as to the likelihood that you have of developing those symptoms over time, when that is used and how, right? And I think there are questions about whether that information, which is actually emerging as a very powerful and potential way of using it as an adjunct to sort of lifestyle changes and understanding our health in general, is how can we use those common genetic changes to understand our risk of diabetes, our risk of different health concerns, right? But the question is, when you're making decisions that are important, when is that relevant or not? And is it relevant for all differences, right? So do you do it for psychiatric differences? Do you do it for issues of identity that have to do with gender health and sort of who we are? Do we do it with in the prenatal setting as your family planning? Do you then say, if I can predict whether my next generation is going to have X, Y, or Z, do I use that information to make decisions about what my next family member is going to look like, right? So I think all of those are big, big questions that are being addressed on the clinical side as to whether the fact that we have this technology means that we should take advantage of it in all settings at all times, right? And I leave to Zach the more research-related questions, which are also very important as well.
Zach, you're up.
Yeah, and I think really when it comes to the autism community's concerns, and one of the major things that's at the front of everyone's minds is what is going to happen with people's research data. Because one thing that a lot of autistic people are really genuinely worried about is the thought that people can use large banks of genetic data to do things like come up with prenatal screening for autism, and then use that to essentially, give prenatal genetic counseling to expectant mothers and say your baby has this likelihood of being autistic if you want to terminate the pregnancy. Now, I mean, that's obviously very controversial and probably, quite unlikely based on how well we're able to predict autism with polygenic scores and things like that. But theoretically, if you are able to, have better predictive models with, your whole genome sequencing and more data, this is at least something that these large banks of data could contribute to feasibly. And so people in the community are worried about the opportunity for some bad actor to at least apply for this data or these data if there are not appropriate protections in place. And so this is essentially what started this whole backlash from the community against this major study, was that there wasn't enough in place to say that this couldn't happen. The research team, clearly came out and said, like, no, we are against prenatal testing for autism. We are against eugenics. We do not want this. But it was apparent that they still were interested in coming up with polygenic scores for other things in relation to autism, because this is part of how like large scale genomics research works, and like understanding the phenotypes of autistic people still comes up with them getting a lot of autistic people's genomes together, which still takes their data and makes it available. And so then how that is safeguarded and the ethical protections of those data, and how to prevent those data from being misused for nefarious purposes or purposes that are antithetical to what the autistic community stands for, becomes a really, really, prime ethical discussion for everyone in the community, researchers and autistic people alike. And those are the things that we're still grappling with. The Spectrum 10 case study, after a 6-month hiatus and redesign with many external consultations with the autistic community, a new community board. I mean, they've done quite a lot to try and bring this back and incorporate new partners, hired an external consulting firm. I mean, I hope they're doing this the right way. I don't know. I haven't actually seen much of how things have gone since this, but with all the press on them, I believe that they've definitely got a lot of time, money, and effort put into doing this right. So I'm really hopeful that they can show people that genetics research can be done ethically and with the proper amount of community involvement and attention paid to these really tough issues, because I think it's important that we still study these topics, but in a way that the community can get behind and be able to work with.
Great, thank you. And then Felicia, as a parent, do you have ethical concerns around genetic testing?
Absolutely. I would second everything that Zach just said, but just from a parent's perspective, that is exactly what we worry about too. How is this information going to be used, and how is it going to be protected, and how will it impact processes in the future? So that is definitely a concern. The other concern that I have as a parent is just how the community is educated about genetic testing, because the simplest things are created and then people are misled, they're misinformed, they get in an uproar, or they have this expectation. And as families, especially our families, like we're out there real researching, we're trying to find the answers, and there are a lot of bad answers. And so I think one concern would be making sure, if this is something we're doing, that we provide adequate educational opportunities and materials and information to our families, so that they have the right information and they are informed properly so they can make, quality decisions about it.
Great, thank you so much. And our final topic is about access to genetic testing and how we can improve accessibility. Dr. Martinez?
So I'll just briefly summarize what Felicia said, right? So what we're trying to do is establish conversations with primary care stakeholders, who are the ones that are actually having perhaps the first point of contact for any individual who's seeking healthcare. I think underserved populations are experiencing a different healthcare system than most others. And I think that's the main concern that all of us have, that disparity that's intrinsic to the healthcare system is further causing barriers to access. So I think that's one that we want to improve, through improvements in primary care providers' education and literacy about genetic testing and communicating that to families, also doing it directly to individuals, autistic or their families, to actually be more empowered, to know more about genetic testing and have access to those resources for self-knowledge and information-seeking individuals. And then, of course, we want to sort of implement processes that reduce the time to wait to see a practitioner, right? To facilitate testing. One of the initiatives is to try to make testing as cheap and as affordable as possible, so that it can be more broadly available to as many people as possible. So the technology side is moving in that direction, and then working with payers to make sure that that is also supported as well with clinical evidence of its utility, right? So I think all those processes in parallel are going to sort of move the needle forward on improving access.
Great, thank you. Zach?
And yeah, well, I think that most of those like healthcare level interventions are going to be the primary way that we work to improve access to genetic testing. I think another thing to think about is in attitudes toward the field of genetics and genomics in general. So it's no secret that the autistic community is very wary of like genetics as a field, just because of the whole Spectrum 10K thing. And I feel like medical genetics, despite being fairly separate from that type of genetics research, has still really suffered a crisis of reputation as a guilt by association. And so one thing that could really be done to help restore the faith of the autistic community, which certainly interfaces with the parents of autistic kid community, would be to have researchers improve their ties with community members and show more participatory research, and that genetics research can be done in a nice and ethically sound way. And so in doing that, the overall attitudes toward genetic testing would probably improve, which would then perhaps make the amount of people offered genetic testing who then go and see a genetics provider go up.
Great, thank you, Zach. And Felicia, your thoughts about how we can improve access to genetic testing?
Yes, I agree with with everyone here. I'll just second what they've said. But one thing that came to mind for me is making sure that the information is available to all cultural communities. I know right now there's just such a barrier to how people get information. And so what happens is those families who are well connected, they are well connected. And then those families who are not, they never know. And so, you know, maybe just not through the primary care, but go to where the people are to make them aware, and maybe have someone that looks like them so that they're more trusting, and they're able to relate culturally to them. That will definitely help with education, and it will definitely help with access, people willing to be open to it, changing their attitudes about it. So I think keeping that piece in mind, it will be very helpful on top of the things that were shared.
Great, thank you. So I want to thank all of our panel members for participating in this ARRB Lend video today. This concludes our video. If you want to connect with any of our speakers from today, please contact us by the email addresses you see here. And we invite you also to follow the progress of the ARRB network on our social handles that are listed here. And thank you again to our panel members, and thank you to all of you for joining us today for our video.
Thank you.